Welcome back, everyone, to the All Sorts podcast, your place for, well, diving deep into the amazing complexity of the world. Imagine this tiny, intricate maze, a dance of molecules that basically runs the whole show of life.
And that dance, that core code is exactly what we're getting into today. We're exploring DNA.
Deoxyribonucleic acid. We're going to unpack its structure, how it copies itself for replication, right? How it fixes mistakes, and how we're actually learning to engineer it now.
Exactly. We've got sources covering everything from, the early discoveries right up to the latest tech that uses DNA.
So our mission for this deep dive, pull out the key info, the really surprising bits about DNA, and hopefully leave you with a clear and honestly pretty fascinating picture of this molecule that's just central to everything.
Sounds good. Where should we start? The structure.
Yeah, let's start there. The blueprint itself, DNA structure.
Okay. So at its most basic, DNA is a polymer, a long-chain molecule made of repeating units. These units are called nucleotides.
Nucleotides, like beads on a string.
Sort of, yeah. But it's usually not just one string. The famous structure is the double helix. Two of these nucleotide chains, these strands, coiled around each other, like a spiral staircase.
A spiral staircase.
Okay.
And they run in opposite directions, don't they? Anti-parallel.
That's right, anti-parallel. One runs one way, the other runs the opposite way, chemically speaking, and that has big implications for how it gets copied later on.
Right. So these nucleotides, the beads, what are they made of? It's not just one thing, is it?
No, each nucleotide has three parts. There's a sugar molecule deoxyribose, hence the name a phosphate group, and then one of four different nitrogen-containing bases.
The bases, that's the letters, right? A, C, G, and T.
Exactly. Adenine, cytosine, guanine, and thymine. ACGT. That's the actual code.
So how do they connect between the two strands of the staircase?
Well, that's the really elegant part. They follow strict pairing rules. Adenine A always pairs up with thymine T across the helix.
A with T.
Got it. And guanine G always pairs with cytosine C.
G with C. Okay. And they're held together by bonds.
Hydrogen bonds. Think of them like weak magnets. A and T are held by two hydrogen bonds.
Two bonds.
G and C are held by three hydrogen bonds. So the GC pair is actually a bit stronger, a bit more stable.
Three bonds. So if a DNA molecule has more G's and C's, it's tougher to pull apart.
Precisely. We call that higher GC content. And yeah, it generally makes the helix more stable, especially against heat.
Interesting. And the sides of the ladder, the backbones, what holds the individual nucleotides together in a single strand?
That's the sugar-phosphate backbone. The sugar of one nucleotide links to the phosphate of the next, creating this continuous chain. The bases stick inwards, ready to pair up.
Okay, structure makes sense. Double helix, anti-parallel strands, sugar-phosphate backbone, and the ATGC base pairs holding it together. But I've also heard about grooves, major and minor.
Yes, good point. Because the way the bases connect to the backbone isn't perfectly symmetrical. When the strands twist, it creates gaps or grooves along the outside of the helix. One groove is wider, the major groove, and one is narrower, the minor groove.
And why does that matter?
Well, the edges of the base pairs are more exposed in the major groove. And this is crucial because proteins, like transcription factors that regulate genes, need to read the DNA sequence.
Yeah.
They often bind in the major groove where they can recognize specific base patterns.
So it's like the main access point for regulatory proteins. Clever.
Oh, very clever. Evolution has sort of made the important information accessible.
Okay. Now, something else. Non-coding DNA. I read that in humans, it's like over 98% of our DNA doesn't actually code for proteins. What's all that stuff doing?
That's a huge area of research. For a long time, it was called junk DNA, which was clearly a mistake.
Yeah, sounds dismissive.
It really was. We now know a lot of it is involved in regulation, turning genes on or off at the right time and place. Some parts are structural, some are involved in chromosome maintenance. It's like the genome's operating system, not just the application files.
The operating system. I like that analogy.
Right.
And the double helix itself, the B form, isn't the only shape DNA can take, right?
Yeah.
The sources mentioned aDNA, zDNA, even quadruple helixes.
That's right. DNA is actually quite dynamic. bDNA is the standard form in our cells, but under different conditions, like less water, it can form aDNA, which is shorter and wider.
Okay.
zDNA is really weird. It twists the other way, to the left. It might pop up occasionally and play a role in gene activity. And yes, quadruplex structures, G quadruplexes, can form in regions rich in guanine. 4 strands. They seem important at the ends of chromosomes, telomeres, and maybe in gene regulation too.
Wow. 4 strands. The structure is more versatile than you first think. And what about chemical changes to the DNA, like methylation?
Ah, epigenetics. Yes, chemical tags can be added to DNA bases, most commonly a methyl group added to cytosine, making 5-methylcytosine. This doesn't change the sequence, the ACGTs, but it affects how tightly the DNA is packaged and whether genes can be read. It's a major way gene expression is controlled.
So it's like adding sticky notes to the blueprint, changing how it's used.
Exactly. And you mentioned other chemistries.
Yeah.
There was that controversial idea a while back about life potentially using arsenic instead of phosphorus in the DNA backbone.
Oh, yeah, I remember hearing about that.
Well, the scientific consensus now is very much that phosphorus is essential for life as we know it. But thinking about these alternatives helps us understand why phosphorus works so well for information storage.
Okay, so we have this amazing dynamic structure, but it needs to be copied perfectly, right? Every time a cell divides. Replication. Absolutely fundamental. And the key concept here is that it's semi-conservative.
Semi-conservative meaning?
Meaning when the double helix unwinds and copies, each new DNA molecule ends up with one of the original strands and one brand new strand. Half old, half new.
Half old, half new. That makes sense for preserving the information. So how does the copying actually happen?
Well, it starts at specific points called origins of replication.
Yeah.
The helix unwinds there, creating replication forks, Y-shaped structures where the strands separate.
Unzipping the DNA?
Kind of, yeah.
Okay.
Then the main enzyme, DNA polymerase, comes in. Its job is to read the template strand and add the correct complementary nucleotide to build the new strand. A opposite T, G opposite C. But you said earlier the strands are antiparallel. Didn't you say polymerase only works in one direction?
Exactly. DNA polymerase can only add nucleotides to one end of a growing strand, the 3' end. So it synthesizes DNA in the 5 to 3'4 direction.
Okay, 5' to the 3'. So how does that work if the two template strands are running opposite ways?
Ah, that's where it gets clever and a bit complicated. One new strand, the leading strand, can be made continuously because its 5 to 3' direction follows the unwinding replication fork. Smooth sailing.
Easy peasy. What about the other one?
The other one, the lagging strand, is trickier. Its template runs the opposite way. So as the fork opens up, the polymerase has to synthesize the new strand backwards, away from the fork, in short chunks.
Short chunks.
Yeah, these are called Okazaki fragments. To start each fragment, another enzyme called primus lays down a short RNA primer. DNA polymerase then extends from that primer, making a short DNA piece. Then it has to jump back, wait for the fork to open more, and start another fragment.
Wow, okay, so it's like stitching it together piece by piece on that side.
Exactly. The RNA primers eventually get removed and replaced with DNA, and then another enzyme, DNA ligase, acts like molecular glue to seal the gaps between the Okazaki fragments, making a continuous strand.
Ligase, the glue. Okay. Leading strand, continuous, lagging strand, and fragments. Seems complex, but it gets the job done.
It does. And the process is incredibly accurate thanks to that base pairing, and also because DNA polymerase itself has proofreading abilities. It can check its work and fix mistakes.
Proofreading. Nice. And it happens fast, too, doesn't it, with multiple starting points?
Yes, on long chromosomes, there are many origins of replication working simultaneously. Multiple forks moving along the DNA make the whole process much faster. And other proteins help too, like ones that hold the single strands apart and protect them during replication.
Okay, so replication is pretty robust.
Yeah.
But DNA can still get damaged, right? Things go wrong.
Absolutely. DNA is under constant assault from, radiation, chemicals, even just errors during replication sometimes slip through. Damage happens.
So what does the cell do?
It has repair crews. There are multiple sophisticated DNA repair mechanisms constantly scanning the genome, detecting damage, and fixing it. Our sources don't go deep into the specific pathways, but the key thing is they exist and they are vital.
To maintain the integrity of the code.
Exactly. Without efficient repair, mutations would accumulate rapidly, leading to diseases like cancer or other genetic problems.
And I think I read that sometimes for really bad damage, like a break in both strands, recombination is used.
Yes, genetic recombination, where bits of DNA are exchanged between similar chromosomes, can be a powerful repair mechanism, especially for double-strand breaks. It can use the undamaged copy on the sister chromosome as a template to fix the break accurately.
Incredible. The cell has all these systems working constantly, which brings us to us messing with it. DNA engineering.
Right, genetic engineering. This is where we've learned to manipulate DNA in the lab. It's been revolutionary.
What are the basic tools?
Foundational techniques include using restriction enzymes. These are like molecular scissors that cut DNA at very specific sequences.
Specific cutting.
Okay. And the polymerase chain reaction, PCR, you mentioned that one earlier.
PCR, yeah. That lets you make billions of copies of a tiny piece of DNA, right?
Oh.
Huge for forensics, diagnostics.
Absolutely indispensable. These tools allow us to isolate genes, modify them, and create what's called recombinant DNA.
Recombinant DNA. That sounds constructed.
It is. It's basically combining DNA sequences from different sources that wouldn't normally be found together. We can create custom DNA molecules.
What do you do with them?
We can put them into organisms, often using carriers like plasmids or engineered viruses. This creates genetically modified organisms, or GMOs.
GMOs, okay. Applications.
Huge applications. In medicine, we use GMO bacteria or yeast to produce therapeutic proteins like insulin for diabetes or components for vaccines. In agriculture, crops can be modified for things like pest resistance or enhanced nutrition.
Right. Lots of debate there, but definitely powerful. What about identifying people? DNA fingerprinting.
DNA profiling, yeah. It doesn't usually look at genes, but at highly variable regions of repetitive DNA in the non-coding sections. Everyone has a unique pattern of these repeats, like short tandem repeats, or STRs.
Unique patterns. So you compare a sample from a crime scene to a suspect.
Exactly. If the patterns match across several different repeat regions, the identification is extremely reliable. It's solved countless cases, even very old ones.
Yeah.
Of course, you have to be careful about things like sample contamination.
Contamination, right? Always a risk. And handling all this DNA data. Sequencing genomes generates massive amounts of information. Bioinformatics.
Bioinformatics is crucial. It's the field that uses computer science and statistics to analyze biological data, especially DNA and protein sequences. Think of the Human Genome Project, sequencing billions of base pairs. You need powerful computers and algorithms to store that, search it, compare sequences, figure out what genes do, study evolution. It's a whole field in itself.
Computers making sense of the code. And it goes even further. DNA nanotechnology. Using DNA to build things.
Yeah, it's amazing. Because DNA-based pairing is so specific, you can design DNA strands that will self-assemble into specific nanoscale shapes, like tiny boxes or lattices. You can build molecular machines.
Molecular machines. And storing digital data in DNA?
That's another futuristic but developing area. DNA is incredibly dense information storage. You could potentially store vast amounts of digital data in a tiny amount of DNA for a very long time. Still early days, but the potential is huge.
Mind-boggling. And one last thing from engineering DNA enzymes. DNA acting like an enzyme.
Right, DNA enzymes or catalytic DNA. These are DNA molecules often engineered in the lab that can actually catalyze chemical reactions like cutting RNA or even other DNA molecules. They have potential uses as diagnostics or even therapeutics.
So DNA isn't just passive information. It can be an active tool as well. Okay, stepping back a bit, how does the information in DNA actually flow? The central dogma.
The central dogma of molecular biology. It describes the basic flow of genetic information. Francis Crick originally formulated it. The common simplified version you hear is DNA makes RNA and RNA makes protein.
RNA makes protein. That sounds familiar.
It captures the main pathway. The first step we already discussed. DNA replication. DNA making DNA for inheritance.
Got it.
Then comes transcription. That's where the information in a segment of DNA, a gene, is copied into a messenger RNA molecule, or mRNA. This is done by an enzyme called RNA polymerase.
So DNA to RNA is transcription.
Right. In eukaryotes, like us, that initial mRNA transcript often gets processed bits get cut out, a cap and tail are added before it's ready.
Okay, processed mRNA. Then what?
Then translation. The mRNA molecule travels out of the nucleus to the ribosomes, which are like the cell's protein factories. The ribosome reads the mRNA sequence in three-letter words called codons.
Codons.
Each codon specifies a particular amino acid. The ribosome reads the codons and strings together the corresponding amino acids in the correct order to build a protein.
So mRNA sequence dictates the amino acid sequence. RNA to protein is translation.
Precisely. DNA-RNA-protein. That's the core flow. But there are exceptions or additions.
Like what?
Well, some viruses, like retroviruses, can do reverse transcription making DNA from an RNA template.
RNA back to DNA.
And some viruses replicate their RNA directly, RNA to RNA. There are also cellular processes like RNA silencing that involve RNA replication. And then there are things completely outside this flow, like how some peptides are made without ribosomes, or prions, which involve protein shape inheritance.
Prions, right. But the main flow is DNA to RNA to protein. Understanding this sequence and the structure took a long time, didn't it? The history is pretty interesting.
Oh, absolutely. It's a fantastic story of scientific discovery. It really started back in 1869 with Friedrich Miescher. He isolated this stuff from the nuclei of white blood cells, called it nuclein. Didn't know what it did, though.
Nuclein.
Then Albrecht-Kossel figured out it contains specific bases, A, C, G, T, though uracil in RNA. Phoebus-Levine identified the nucleotide unit, sugar, phosphate, base, and the deoxyribose sugar specific to DNA. But people still mostly thought proteins carried heredity.
Because proteins seem more complex.
Exactly. Though some, like Nikolai Koltsov in Russia, had early ideas about a giant hereditary molecule. A key step was Frederick Griffith's experiment in the 1920s, showing that something from dead pathogenic bacteria could transform harmless bacteria into deadly ones. The transforming principle.
Something was transferring information.
Right. But the definitive proof that DNA was that something came from the Hershey-Chase experiment in 1952. They used viruses that infect bacteria and labeled either the viral protein code or the viral DNA with radioactive markers.
Then what happened?
They found that only the labeled DNA entered the bacteria and directed the production of new viruses. The protein coat stayed outside. That pretty much sealed it. DNA carries the genetic information.
Hershey-Chase. Okay, crucial experiment. And then the structure. The double helix. This involves Rosalind Franklin, right? Her X-ray images.
Absolutely critical. Rosalind Franklin, working with Maurice Wilkins, produced incredibly clear X-ray diffraction images of DNA fibers. One image in particular, photo 51, was basically the key piece of evidence.
Photo 51.
It clearly showed DNA was a helix and provided crucial measurements about its dimensions. James Watson and Francis Crick saw this data, perhaps controversially at the time how they accessed it, and combined it with other knowledge, like the base pairing rules, which were being figured out by Erwin Chargaff.
Chargaff's rules, equal amounts of A and T and G and C.
Exactly. Putting it all together, Watson and Crick built their famous double helix model in 1953. They even announced it informally down at the Eagle Pub in Cambridge.
Amazing. A pub announcement for one of the biggest discoveries ever.
And things moved fast after that. Crick formulated the central dogma in 1957, and in 1958, the Mendelssohn-Stahl experiment brilliantly confirmed the semi-conservative replication mechanism we talked about earlier.
Mendelssohn-Stahl.
These discoveries together really launched the era of molecular biology, understanding the code, how it's copied, how it flows.
It really is an incredible journey, from isolating nuclein to building molecular machines out of DNA.
It truly is. The elegance of the structure, the complexity of its replication and repair, and now our ability to actually engineer it. DNA is just this molecule of profound importance and, frankly, beauty.
It makes you think, doesn't it? What are the limits? Understanding and manipulating this code.
Right.
What does it really mean for medicine, technology, our understanding of life itself?
The journey is definitely not over. The potential is still unfolding, and future research will undoubtedly bring even more mind-blowing insights.
Absolutely fascinating. A huge thank you for walking us through that. And for everyone listening, if this deep dive into the code of life sparked your curiosity, please do us a favor like this, share it, and subscribe to the All Sorts podcast right here on YouTube.
And don't forget to check out our back catalog. We have loads of other deep dives exploring all sorts of fascinating topics.
That's right. And to keep the conversation going and stay updated, find the All Sorts podcast on X, Instagram, and Threads. Follow us, join the community. Let's keep exploring the wonder together.
Thanks for tuning in.